does our brain communicate with our lungs? How does our blood communicate with what is being filtered out in the kidney? So these are the questions that would come to our mind whenever we talk about the language of life. And in this webinar, I'm going to answer some of the peculiar questions that regards that particular topic. The name is Dr. Ashraf Akintola, and here's a webinar with the title, The Language of Life in Data Form, Gene Expression and Biological Process. And here we'll be talking about the language of life in data form, that is, in data form that we can understand, in data form that our body speaks that we can understand, how our body communicates. And here we'll be talking about the gene expression, transcriptomics, and how our body decodes all the biological processes and how we are going to decode the biological processes that goes on in our body. So there's a saying that goes, if DNA is the script of life, then gene expression is the performance. If DNA is the script of life, gene expression is the performance. Now, I want to welcome you to this. And why does this topic matters? Now, today we answer these questions. Number one, how does the cell know what to do? How do genes in our body turn on and turn off? How do scientists listen to what the cells are saying? And where would you start from if you are a beginner that wants to venture into this session or into this knowledge? Let's take the questions again. How does the cell know what to do? How do genes in our body turn on and turn off? And how do scientists listen to what these cells are saying? And where do you, as a beginner, would you start from if you also want to be among those scientists that listen to what the cell is saying? So this session is poised to give us clarity, direction, and confidence, especially if you are starting your bioinformatics journey, if you are transitioning into genomics or precision medicine, if you are trying to understand what gene expression data means, and if you are looking for the right tools and courses to follow. So these are what this session will give you clarity on. Now, we have to understand that life speaks a language. Our body speaks a language. The living things in our environment, even we ourselves, our body speaks a language. Most times, the language that we do not understand. And this language is not English, it's not Yoruba, it's not Arabic, it's not Korean, it's none of the languages that we human beings speak with our mouths. But cells communicate through special molecules, and these molecules are categorized into different divisions. Number one is the DNA, which is the instruction manual that gives the body everything it has to do. Everything that this body has to perform the function, it's already written in the DNA. How would you read the language of the DNA? How would you interpret what is inside the DNA? How would you know what the cell is communicating to each other through the DNA, the instruction manual that is unchangeable? Yes, in some aspects, we're not going to go into much details into that later on. Now, we also have the RNA. The RNA is ribonucleic acid, and which is the messenger that make sure that the instruction manual, the instructions that are written in the DNA, which is the instruction manual, are taken to the right direction. And then the proteins, which are the workers. So these are molecules with which the cell communicates with. Now, if you are able to read the DNA, the RNA, and also the protein, you'll be able to understand the language of the body, any living body that produces these molecules. So now, a simple definition of gene expression is now how the cells in your body turn on or turn off the genes. Now we know that the genes are the single unit of single functional unit in your body. For example, there is a particular gene in your body that is called insulin that is used for sugar conversion. That is a particular gene in your body, a protein rather. There's a particular protein in your body that is called insulin. And this insulin is also gotten from a particular gene. So we're not going to get into much details regarding that. So this gene are the singular functional unit in your body. And how do they turn on and turn off is what we know as gene expression. So an example of that is when you are stressed out, there is a stress response genes that are turned on. When you want to eat, insulin producing genes are turned on. Insulin producing genes, I mentioned earlier on, as insulin producing genes are turned on when you eat because they are responsible for converting sugar to what the body can store. So insulin producing genes are turned on. So that means there are genes that are responsible for producing the protein called insulin. So now when viruses invade, immune genes are turned on. So these are what we call gene expression, how the genes express themselves. So when cancer forms invade your body, then when cancer forms, that is when cancer in a particular cell in your body forms, when they start to grow, then cell cycle and growth genes become abnormally on. There is a way in which cancer, when they say something is a cancerous growth, that means it's an abnormal growth of a particular cell in your body. So when that happens, you will notice that there will be an abnormal growth in the cell cycle and growth genes in your body. So that means gene expression is the real-time activity in the cell of a particular entity. So what does this gene expression, what does it tell us? Gene expression tells us a lot of things, but among them, it helps us to understand why diseases happen, why drugs work or fail, and how cancer evolves. It tells us how immune cells respond, and it tells us how tissues develop in our body, and it tells us how organisms adapt to change. And that is why transcriptomics, which is the study of how these genes are expressed, is a core to precision medicine, it's core to drug development, and it is core to disease modeling. If you want to go into these three different areas, precision medicine, drug development, and disease modeling, that means you have to be an expert in transcriptomics. Now, here is a biological workflow of gene expression. That is, one, the DNA is the instruction book. We've mentioned that earlier on, which is the instruction manual. We have transcription, which is the RNA, how the DNA converts or changes into RNA, which are the messengers, and then how the RNA translates into protein, which are the ones performing the actual work. Now, the biological process will now lead to cell functioning for metabolism, for growth, and for immunity. And then we have the next stage, so which progresses. Now, what does gene expression data look like? For those who are just beginning, it might look very gigantic. It's a giant spreadsheet. Just imagine it as a giant spreadsheet that contains rows, that contains columns, and that contains number. The rows are responsible for the genes. The columns are responsible for the samples, and the numbers are responsible for the expression levels. As a beginner, that's what you will see. But for experts, you'll see what we call the counts. The counts is the term that is being used for gene expression. The gene expression count. You have the TPM, we have the normalized values, you have the differential expression statistics. So a gene expression is the total number of numbers that represent a gene activity. So there's a special number allocated. Okay, this is a special number that's allocated this particular gene at this particular point in time. Now, moving forward, what are the most common questions? To answer the most common questions that some of our students are asking us, that how do we know which genes are active? Of course, it's very, very simple. We know the genes that are active when we measure the RNA abundance. Don't forget the RNA, they are the messengers. We measure them, we measure how genes, we know which genes are active when we measure the RNA abundance. So the more it is abundant, we know that these are the genes that are presently active. Now, what does gene expression data even look like? Of course, I've explained that. It's a rows and columns, that is a matrix of genes and samples measured by sequencing. And where do I start with transcriptomics? You start with transcriptomics by learning the workflow of the process itself. So transcriptomics, which is the study of the RNA molecules produced by a cell or a tissue. So we've told us that it answers which genes are expressed, how much they are expressed, and how do they change across conditions. That is, if a tissue or a cell is healthy and if a tissue or cell is diseased, you will be able to infer a difference between the two of them, that okay, these are the genes that are expressed in this one and these are the genes that are not expressed in this one. They call it upregulation and downregulation of genes. Now, these are the simple definition and the simple steps that you have to understand before you say you want to venture into transcriptomics. There are different types of transcriptomics. You have the bulk RNA sequencing, we have the single cell RNA sequencing, and we have the spatial transcriptomics. So these are the three ones. However, right now people are moving on to single cell RNA sequencing. That doesn't mean that you do not have a lot of work to do in the bulk RNA sequencing, and then you can move on to the spatial transcriptomics, where this one will show you where the genes are active inside the tissue. So that's the types of transcriptomics that you have. Now, to briefly take us through the gene expression workflow as a beginner, the first step is the experimental design, where you take the control and versus condition tissues and replicate the control, the condition, the control, and the disease condition, and then you replicate all those samples. The step two is the RNA extraction. You extract from the two of these samples, and then you sequence, of course, through FASTQ, and you get the FASTQ files. From the FASTQ files, you do what we call the quality control, which is the step 4, which is the FASTQC and the multi-QC. Step 5 is the alignment. Step 6 is the quantification. Step 7 is the differential expression, where you are going to be studying the differential expression in these particular genes. So, and then you'll be using resources like the seq2 and EDGE-R, which are present in R. And then you have the step 8, which is the biological interpretation. We'll be using GO, the gene ontology, the KGG, the GSEA. All these are the simple steps that you have to go through, the 8 steps to go through when you want to do transcriptomics analysis. So now there are different tools that you can use as an expert or as a beginner, and that will be taught to us in our bioinformatics diploma course, and which I'm going to invite you to join in our next cohort. The time and the date will be displayed to us and will also be shared to us in this video, in subsequent announcement. So we are having a present cohort going on presently, and then the next cohort will be next year. So we'll be looking forward to you joining us on how to understand the languages that the body speaks and the languages that life speaks itself, so that we can provide solutions to the problem that confronts us as human beings in medicine, in agriculture, in environmental sciences, in virtually all the sciences that deals with life. Anything that life form is involved, definitely you'll be needing to study this particular area of sciences. Now, biological processes explains how we've understood these biological processes and how data experience life. Transcriptomics reveals the pathway activation, the cell cycle progression, the immune response levels, the metabolic activities, the cell signals, the drug responses, the cell differentiation, and it will definitely tell you that every biological process leaves a data signature. There's a data that follows every biological process that you can also work on. Now, here are examples of hand-on examples of how you can utilize transcriptomics in your day-to-day activity. Number one, example one is in cancer. You can identify genes that are upregulated in different cancer, in tumors, maybe in brain tumor, in ovarian cancer, and the likes. So you can identify genes that are upregulated. These upregulated genes will definitely be your target if you want to do what we call the drug development. I told you earlier on that if you want to go into precision medicine, in drug development, and into disease modeling, you have to know transcriptomics. And this is one of the steps that you pass through. Another example is in infection. You can see which immune genes turn on during viral infection. Which immune genes turn on during viral infection. So that is for infection. So immune genes that turn on, that means these immune genes are the ones that are actively working against this particular infection. So you can boost that the genes enable to for the body to naturally withstand that. Number 3 is drug response. So find genes affected by treatment. You can find the genes that are affected by the treatment. So using the method that we've discussed earlier on. You can also, in example number 4, brain tissue, you can identify neuron versus the glial cell types. So these are some of the myriad of examples that you can get to do with transcriptomics. Now, how do you as a beginner, yes, I'm assuming you're a beginner, how do you as a beginner start? There's a clear roadmap for you, and it is a 6-step clear roadmap. Number 1 is for you to learn the basics of RNA and gene expression. That is the basics of RNA and gene expression from DNA to RNA to protein. Understand that. Step 2 is understanding transcriptomics workflow. Understanding transcriptomic workflow. We've explained it earlier on. Number 3 is start with accessible tools. All these tools gladly are readily available. You have Galaxy, you have RStudio, we have Python notebooks. Start with accessible tools. Number 4 is practice with real data sets. There are lots and lots of data sets that you can practice with if you want to start on your journey into transcriptomics. Step number 5 is to learn differential expression. Differential expression, GO, GSA, HR, DSIC2, and the likes. And then step 6 is to move into advanced techniques. That is, if you want to do bulk RNA, you want to do single cell RNA, you want to do spatial technology, spatial analysis. So these are some of the ways you can progress. The steps 1, 2, to seek the step number 6. Now, what are the career pathways that you can choose if you are going into gene expression and transcriptomics? Number 1, of course, as a bioinformatician. Number 2 is the clinical genomics analyst. You can be a single cell data scientist. You can be a precision medicine researcher. You can be an academic researcher. You can work in the pharmaceutical companies or biotech R&D. You can also work as a genomics consultant. So, and this is because the gene expression study is the future. Because gene expression is real time, it is condition specific, it is highly sensitive, it is biologically meaningful, it is predictive of treatment response, and it is central to precision medicine, central to drug development, and central to disease modeling. So now these are some of the things that you will learn from our bioinformatics diploma program. So join us in our next cohort, and I'm willing and wishing to see you soon. And I want to thank you for joining this particular webinar. And when we code gene expression, we understand life itself from our short communication, from our short moment together. Thank you for attending. Now let's take your bioinformatics journey to the next level. See you in another webinar. Thank you.